When we got that data back, I think there's always celebration, but there's a lot of relief. And that what you've done is you've been able to demonstrate that without any, pretreatment, you've been able to get a clean signal. That clean signal now is something you can build on top of.
Welcome to the Labs Report. I'm Justine Levin-Allerhand. I'm back here today with Jagesh Shah, Senior Vice President and Head of Platform at Mirai Bio. Welcome, Jag.
Thank you.
So you've been part of the Flagship ecosystem now for a number of years, first at Cobalt Biomedicine, VL39, and then Senior Fellow of Sana, and now Mirai, formerly known as FL85. So your career journey actually is a great example of what it means to be part of the Flagship ecosystem. You start in one company, you have major impact, then other opportunities emerge and there are synergies, and you move to a different company. So you've been in a few different companies now at Mirai Bio, but there is a connection in this case where all three of them were in pursuit of in vivo genetic medicines. So eager to hear a little bit more about your journey and hear about what your role is at Mirai Bio currently.
So thank you for having me, Justine. I'm very excited to be here. So Mirai, as I think we'll discuss, is part of a new group of companies at Flagship where we're not just innovating, but we're trying to partner out that innovation. So it does change my role a little. As head of platform, I'm still driving the innovation. I have a great team that's excited. It's not hard to recruit, great scientists to Flagship to do innovative science. But we also spend a lot of time pitching to partners, partners that have nucleic acid cargoes that we want to put into our delivery vehicles. So it's an exciting opportunity because we get to innovate our own science, but then we get exposed to a lot of different kinds of sciences in a lot of different companies. So that's a lot of fun.
Yeah, it's great. What's interesting about that comment you highlighted, you're talking to a number of partners, they've got interesting cargoes and you have to use the vehicles that you're creating at Mirai Bio for delivery. And for me, that's a conversation that I feel like I've been in for over the last 10 years, dating back when I was at the Broad Institute and when there were just amazing strides technologically in genome editing. The conversation was always like, great, how do we actually get them to the actual cell type that we need to have of therapeutic value in time? And, or take antibody drug conjugates, ADCs. Right. ADCs like a mailing address. It's great to get to a zip code, but then you actually need the street address to get to the exact house to bring your package, and the zip code is not enough. So why has delivery been such a challenge? If I'm recalling 10 years ago, you're saying this is something that you're engaging many partners. What's been so challenging? And why a decade later are things different? What's Mirai doing in this space, and what's changed?
Yeah, so I think there's always been an exciting opportunity that being able to reprogram cells with their language is kind of the core element of any genetic medicine. But getting it to the right cell type has been challenging. And so one of the critical inflection points is the recognition that we could deliver nucleic acids and make them less immunogenic. So that was the basis of the Nobel Prize for Weissman and Carrico. And so once we had that, we actually had probably 6 decades of lipid nanoparticle discovery that was just ready to pounce on that. And it turned out we had a pandemic. So now not only did we have the inflection point and be able to use these mRNA technologies for the vaccine, but then we also had to scale. So it's almost like doing drug development in kind of warp speed. All of a sudden, we had, I believe it was called warp speed actually. But the idea that we were able to get not just the early views, we would get human data and we were able to scale. So that I think is critical. The other piece is now the linchpin of any LNP is the ionizable lipid, and that's a chemistry space. That can be vast. So how do we navigate that space? And I think this is where ML and AI is a particular tool that Mirai has brought to the table to try to navigate that very, very quickly. So I think that the two pieces is really why Mirai Bio came into being when it did.
So we'd love to double click on the ML, the machine learning side. It seems like the ML, as you said, there were a couple of pieces, but the ML is not just a tool for Mirai. It kind of gave us the confidence that it was a moment in time that we should go after, and we formed FL85 at the time, now known as Mirai Bio. So how has ML been both central to Mirai's value proposition, and as it continues to develop, as you think about the next value inflection point, it seems like doing things in vivo gives us the confidence to keep going and to be engaging with partners.
That innovation of ML application to chemistry is not new. But what the key insight from Mirai was that we were not going to build this on an edifice of in vitro data. In vitro has been valuable for a variety of different technologies, but it wasn't going to give us the understanding of how something biodistributes. If you need biodistribution, you need a circulatory system, you need organs for them to read out. So we chose to rapidly move with a barcoding technology to rapidly get a lot of in vivo data and then use that as the basis for the ML. And I think that that's really the key. So, like all good companies have Flagship, there's a few key technologies that you require to converge. And I think that that's what distinguished us from, and we've already had some early wins, the ability to be able to now take that basis and then have ML guide us to a formulation or a chemistry that gets us to a place that has been challenging to get to. That's already played out. So we're very excited about that.
Yeah, the other aspect that I have been found so interesting is that the ML is not just enabling the design of the LNP and getting to the right cell type, but it's also there to help drive to make sure you're going away from off-target tissue. So you want to be on target and then ensure that you're avoiding off-target. So, in this, one perfect example, one great first example is in T cells, where you're actually putting that to the test. Before we get into the data, share a little bit around why T cells, where the application for those who may not be, in the space of T cell biology and T cell medicine.
I think T cells have been a remarkably unique opportunity to reprogram the immune system and in many cases go after now cancers. So, and the first chimeric antigen receptor T cells were developed to go after B-cell lymphomas, and we've seen remarkable opportunities there. A lot of that is done what we call ex vivo, where you take the cells out, reprogram them, and then put them back in again. The ability to do in vivo CAR T therapies is dramatic. You don't have the same manufacturing challenges. In principle, you could deploy this in a doctor's office. And so that would be a remarkable change. And so we're very excited about this. Frankly, this was a technology that we also developed at Sana in a related way. So LNPs, I think, present a unique set of opportunities as well because they can be reprogrammed so quickly. You just have to change the cargo and you can do something else very rapidly. So the speed of development of an LNP, I think, does have some advantages over some of the other approaches.
The other aspect when making medicines is you always want to be sure that whatever medicine you're making is tolerable. And obviously you have to test that in animals before you go to humans. And especially with LNPs, making sure that whatever you're giving the body, it actually tolerates. So in this case, demonstrating clear tolerability in a non-human primate, which is a critical step in the drug discovery and development pathway often, is really a major milestone that Mirai was able to achieve. Was this expected? It's such a critical part, so we'd love to hear about that.
I think that people always ask, when do you know? Well, strictly speaking, you don't know until you get into a person, but you can get high confidence as you go along. And I think that the NHP or the non-human primate is really the critical stage. And right now, we don't have a lot of examples of LNPs in NHPs. So still that machine hasn't been able to learn tolerability. So this is an area where we're still empirically assessing. And we have a great translational team at Mirai, and we all agreed that we wanted to go in without any kind of pretreatment, just to get the cleanest signal. What the term that we use in-house is we go in neat, like scotch, no ice, right? And you go in, and when we got that data back, I think there's always celebration, but there's a lot of relief. And that what you've done is you've been able to demonstrate that without any, pretreatment, you've been able to get a clean signal. That clean signal now is something you can build on top of. If you get a clean signal once, you can start multi-dosing. You can do a lot of things. It's a gateway to improving efficacy in a way that you have more confidence.
Right, and going back to earlier in our conversation, if you then feed those data back into your models, then all of a sudden you can continue to optimize. So it's a great starting point.
It's an absolute, and I think that's something that perhaps ML forces us to do, unlike any other time in science, is collecting data so that we can reuse it. And, I think in the past we just get the answer and we're like, awesome, we got the answer, and then we move on. But here you can feed it back into
What
the
I would,
machine.
I would argue as humans, we are actually doing that algorithm in our heads and then creating the next experiment,
Actually,
and ML
that's
is giving
absolutely
new
true,
insights.
yeah.
But I agree with you. It
Yeah,
really
yeah.
is
Well,
greater
it
use of the
does
data.
have a capacity to store a lot more information than at least I can
Right,
store. So
exactly.
that is that's absolutely true.
So modularity, or being modular, is a word that is used a lot in biotech as we think about making programmable medicines. And the deep dive actually goes into that aspect of what you're doing at Mirai. The same base particle can reach a T cell as a NK cell, a B cell, and you just swap out certain elements, in this case, a targeting moiety. So tell us more what that means in practice as you, again, start building out more and more delivery vehicles.
I think that gets to perhaps a traditional approach in LNPs, which is bespoke generation of LNPs for almost every application. So starting from the chemistry all the way up, and the ability to go back and reuse information has been challenging. So one of the things we did with the ML is how could we get to a number of tissues that would be valuable with a one LNP. That one LNP we could test in NHP, so we could not worry about tolerability. Mm-hmm. We could also detarget it from the liver, and that would act as what we call kind of base camp. You don't have to go back to sea level every time. You don't have to scale. So that has two factors. One, it increases confidence in the drug you're about to make, but it also allows you to move very quickly. And so the speed of development here is, I think, something very exciting for LNPs. And, being able to get to all those different cell types just by switching out the binder, I think, is something that's been very appealing for both our partners as well as, other groups that we've talked to.
So, Jag, if Mirai Bio does all the things that we discussed, address the tolerability question, ensure that there's proper tissue targeting, and solve the manufacturing needs for these novel LNPs. And if those are all addressed at the platform level, and that's done in a modular way repeatedly, what does that open up for therapeutic programs? What disease can we now go after? What questions are we able to answer?
So we're very excited about T cell targeting. I think that there's still a unique and opportunity there. But I think, as we described in the modularity, now we can start talking about what we can do in these other cell types. I think HSCs, there's an ex vivo to in vivo playbook that a number of companies are moving towards, and I think we can really enable some of those activities. But B cells, NK cells, these are cells that, we are trying to reprogram as well. We know that CAR NKs have been used in autoimmune disease as our CAR T. So there's just a whole host of activities that we think that this can unlock. Probably one of the most exciting things is getting to these other tissues and unlocking tissues like the pancreas or the kidney or muscle or CNS. Those are areas where the ability to be able to deliver, say, a gene editor to the kidney, the gene editor to the CNS, I think is going to be a remarkable opportunity for not just Mirai, but I think just human health in general. So we're excited about the future, and I think that the ability to move fast is really the key, and ML has been critical in generating that kind of data.
So it's really clear that Mirai Bio isn't just solving the delivery problem. They're kind of rewriting the rules of how to think about nucleic acid medicines, which really then opens up a whole new toolkit to going after disease. So much of what we do here at Flagship is pioneering science, where we're going after completely uncertain, unknown spaces. And that process of uncovering that science and then reducing it to practice and applying it is what we do every day. And that's what you're doing at Mirai Bio. So any advice you can give to our pioneers at Flagship, but more broadly across the scientific ecosystem of trying to really uncover new biology and new discoveries that you could apply in real world situations. What's that process like, and what advice do you give in that uncertain space?
One of the principles I hold to, early on is that you do want to swing for the fences. You absolutely want to try to do the most exciting thing that you possibly can. But you don't want to break any laws of thermodynamics. You want to be able to stay within there, and negative results are valuable. Mm-hmm. Negative results help guide you so that you can really focus on the things that matter. I think that personally, this idea of platform science is just invigorating. Not enough people do it. The idea that you really try to understand how to solve a variety of problems at the same time, you can only learn from each of those. And if you can have the right people then you can do anything. So it's really about creating a culture where people are excited about what they learn, whether it's negative or positive, and how do they move forward, and how do you make sure that people really have their eye on big science, because that's what we're here for.
That's great. So what I'm hearing is learn from every experiment. You always have opportunity costs, so choose the right things to work at and have the right team to be on the journey with you on it.
Absolutely.
And eager for everyone to see the full data set and head to our Substacks, the labs reports. So thanks again, Jag. It's great to have you.
Thank you for having me.